Let's try to run through how to design primers for reverse transcription. Here's our message. It's a single-stranded piece of RNA. The 5' end is here. The 3' end is here. Obviously, we're going to design primers that are going to clone this whole thing. But let's start with the one that makes the most sense first. Remembering that if we try to put a primer here, the primer has to lay down anneal in the 5' to 3' direction. So to be complementary to this message, the primer has to look like this. Now, obviously, when reverse transcriptase lays down, there's nowhere to go, so nothing happens. Instead, let's start with a different primer, complementary to the 3' end, like this. But this primer again lays down in the 3' to 5' to 3' direction. This is complementary to the 3' end of the DNA of the RNA. But the way we write this thing down is important for ordering, because there is a directionality here. So if the gene looks like ATCG, if the message looks like ATCG at the 3' end, this primer is going to look like CGAT. But we have to write that down properly, right? It's got to be 5' to 3' to order. So we have to write the primer down, CGAT. That's primer 1. Now, so here we are, our messenger again. And I'll use red for primer 1. In the first round of this process, the reverse transcriptase is going to make a strand of DNA priming from here, the double-stranded region, to make a DNA strand. So we have a DNA strand now and an RNA strand. Now we're going to go through another round of PCR. Actually, the first round of the polymerase chain reaction. The first round was reverse transcription. And I'm going to write it this way. We're going to denature. So there's our original RNA. And now we have our new strand of DNA. And we have our primer number 1, ready to do another round of replication of that piece, amplification of that original RNA. Now we need our other primer. The other primer, I'm going to draw in blue, is going to be complementary to this strand. Remember now, this new strand of DNA looks like this. Oops. Let me try to use my right color scheme. It's going to look like this. 5', 3'. So our primer that we design is now basically the 5' end of the RNA sequence, exactly the same as that. Now it'll just be DNA. So we'll write it down this way, 5' to 3'. And I think you can see that it's going to look just like the sequence of the message itself, but to be complementary to the 3' end of the antisense strand. So let me hopefully that's really clear that we have here the RNA, original RNA, is our sense strand, and the new DNA strand is an antisense strand. So if we want to write our primer down, it's really just going to look like this sequence. So if we write down the 5' end of the RNA, let's write it down this way, ATGC. Then the primer that's complementary to the antisense strand is just going to be the same thing, ATGC. Okay, so that's the primer design. We've got primer 2 that's complementary to the 3' end of the antisense strand, and we've got primer 1 that's complementary to the 5' end to the 3' end of the RNA strand. In terms of our gene, let's remember too that we found 2 pieces, our exon here and our exon here, and we found that the start codon was somewhere about 60 or 70 bases into the first exon, and the stop codon was here, somewhere near the end, maybe even at the end, I don't remember, of the second exon. So this will be primer 2, only used in the second step, and this will be primer 1. Remember,